All right. Hi, everyone. My name is Thomas Werner, and I write fruit fly guides for North America. And I'm going to show you today how I do this when I can't image them in my home lab at Michigan Tech. All right, next slide. So the scientific value of fruit flies is enormous. For the last 120 years, people have done so much research with one fruit fly, Drosophila melanogaster. That is the one that you find in your kitchen that annoys you usually in summer and fall. And with Drosophila melanogaster, you can only do so many things, although you can study human diseases. But there are many other fruit fly species out there that have traits that Drosophila melanogaster doesn't have. So for instance, toxin resistance. Some fruit flies can eat deadly poisonous mushrooms, such as the death cap. Other fruit flies have nice color patterns, and they can be used to study climate change adaptability. So the new genomic tools that we have now, like CRISPR-Cas9 genome editing, and also single fly whole genome sequencing are some fantastic tools that we can use actually in order to study fruit flies that are not model organisms. Next slide, please. So what we need to do is we need to go out into nature and grab that huge diversity of fruit flies and bring them into the lab. And what you see here is on the left, there are some fruit fly baiting sites. I'll show you them in detail a little later. In the middle, you see a couple of collection vials. And then on the right, you see a part of my fruit fly research lab where I have like lots of fruit fly strains. Next slide, please. So if you want to know what fruit flies are in the Northwest of the United States, then you have to take this book from 1921 that is over 100 years old, and it only has 3 images in it. So you will not be very lucky to identify anything, no matter how good you are at catching diversity in the Northwest. And there are 5,000 species worldwide, and in North America alone, there are more than 1,000 species. So I thought, I'm going to change that and write an encyclopedia of fruit flies for North America. Next slide, please. So I already published 2 books. The left one is the volume 1 of my encyclopedia, the Drosophilids of the Midwest and Northeast in its 3rd version. And on the right, you see the Drosophilids of the Southeast, which is also a book. Each of these books have about 70 species in total in each of them. Next slide, please. Here I show you what I think a fruit fly identification guidebook should look like. I usually take all the legs and wings off so that you can actually see the features, like on the left of Drosophila susukii as an example. You see on the top a male with all the key features that you need to know in the current area where you are collecting. On the lower left, you see a female of that, and only the key characters that you must look at, and not more than that, when you are, for instance, in the Northeast, right? And then in the middle, you see the whole variation of 5 males and 5 females underneath. And then on the right, you see kind of like a red, yellow, green traffic light. This is what goes through my head, the 3 key characters in that order, in order for me to figure out what fruit fly species I have in front of me. Okay, next slide. So I want to write more books. I have covered the eastern half of the United States. The next goal is the Northwest. I image my flies alive, and in order to image them alive, you've got to actually have the flies catch them and image them right away. And if you go to the Northwest, coming from Michigan, it is very far. That's why actually I decided to take my fruit fly lab with me. Because otherwise I can't image flies. So I have to image the flies on the road. Next slide, please. So this is basically 2023. We were just going out to the Northwest. On the left is the Michigan Tech minivan with my mobile fruit fly lab. There are my kids also. And then on the right, you see our family minivan, and I go on trips with my family. Next slide, please. And so this is the minivan with the fruit fly lab. Everything is nicely organized there. Next slide, please. And this is the kids, some of them at least. It is not that organized always when you have kids in the car, but fortunately it's not my research van. Next slide, please. So this is what we did last year. We started from the top right corner, Michigan Tech, basically the Upper Peninsula of Michigan, and traveled to the west. And wherever you see a net, that's where I collected fruit flies for a week. Next slide, please. And this is my fruit fly lab. There it is. That's all I need. 2 free little baggies and a picnic table. Next slide, please. And this is when you unpack the bags. There is my mobile fruit fly lab, even with solar power. Next slide, please. And I can gather 400 watt of solar energy, and I have 3 jackeries. I have basically a capacity of 3.5 kilowatt hours. At any time, I can power all of my appliances, my family and my kids' iPads included, and even everything a camera team needs, because I was accompanied by a camera team who made a documentary this past year with me. Next slide, please. So this is basically where we went to Olympic National Park, one of our stops, where the red arrow is on the left. That is Fairholm Campground. And on the right, kind of, you see a picture of what it looks like when you look over Lake Crescent from that corner. And I picked out the site because I needed to find one special fruit fly species. And I kind of figured out this is where I would find it. Next slide. So what you do is you put out fruit fly baits. On the upper left, a banana bottle. There is banana mush in it. On the lower left, you have rotten mushrooms. On the lower right, you have watermelon rind. Next slide, please. That is me collecting fruit flies from a ground bait. There is lots of fruit flies already in that net. Next slide, please. Here are the fruit flies in the collection vial, a mixture of good stuff. Next slide, please. This is my favorite fruit fly, Drosophila flavopinicola. It has the effect on me as if I drink 5 espressos at once. Next slide, please. Drosophila flavopinicola is here under the microscope. This was the first specimen I ever caught, and that was Olympic National Park. There have only been 8 fruit flies of that species being found in the last 100 years. Next slide, please. And this is my fruit fly lab from the inside. And what you see there on my laptop is basically Drosophila flavopinicola. I found a couple of more of these species this summer, and I was able to complete actually all the images that I needed. On the far right, you can see what my lab looks like at night. It's nicely, I have lamps in there and can walk day and night in this lab. Next slide, please. This is the laptop. These are the images I actually took in my tent lab, and they are publication quality. This is Drosophila flavopinicola, my favorite species. Next slide, please. And I finally finished my chapter for Drosophila flavopinicola for my next book, which is not published yet. Next slide, please. The remnants of my flies, they go all in ethanol, and then I have some friends at Stanford University who sequence every individual fly. They do whole genome sequencing, single fly, which is absolutely amazing. Next slide, please. This is basically my summary of the species that I found, all the red dots. You can see I found 11 fruit fly species at Olympic National Park in 2 years, and all the other species around that don't have a red dot. This is kind of the table of contents for my next book. So I'm expecting there to be about 80 species or so. So I found about 11 out of 80 species at Olympic National Park. Next slide. So as I told you, I had a film team with me this summer. They were following us through Oregon and California just after we left Olympic National Park. And there were a couple of students from Stanford also. They wanted to learn from me how to collect fruit flies in nature. Next slide, please. And this is my acknowledgement slide. And on the right side, you can actually see I already picked out the cover for volume 3, Drosophilids of the Northwest. That is my next book. And I would like to thank the NSF and then my Stanford people, Bernard Kim, Dmitry Petrov, Daniel Shaikevi, and Messi Fisher. The last two they have actually filmed. This is the film team. And I want to thank Matthew Dubois, the research coordinator, who helped me get my permits. And I want to thank my family, my wife Megan and my daughter Natalia, and my sons Oliver and Oscar Werner. Thank you very much. And I think I'm on time here. Thanks.
Yes, you are. Thank you, Dr. Werner. Any questions? Carla Cole wants to know why flatwing cola is your favorite species.
So Flavo penicola is, I read about it before I went on my first trip to the Northwest, and I thought to myself, I will never find this one because it's so rare. And then when I saw it the very first time, I actually almost fell backwards from my picnic bench over the microscope, and I really had to catch my breath because I thought I had it, and then I spent like hours looking for the literature, and I knew I had it, and it was like on this trip, I just found like half of the flies that have been found in the last 100 years. And and it's beautiful. So it's kind of like a very emotional moment. When I see these flies, I know I have something really special. You know, you learn to love the flies.
Wants to know how large are fruit fly ranges and how did you know where to look?
So you can look pretty much everywhere in your kitchen. So the size is about 3 millimeters on average, 2 to 6 millimeters approximately. So it's very, very small. And I usually look at forested areas, and I also look where there is either rivers or lakes nearby. So they like to be near water, but in the forest, you get the greatest diversity in those places. Yeah.
And Elisa wants to know, are there any species you want to collect but that you haven't been able to?
Yeah, there are many species out there. I actually would like to catch the one that I named after my student, Tessa Steenwinkel. I only found one specimen in my life, and there are only 4 specimens known on Earth. I would like to go back out there where I caught it, and I would like to put some traps into the tree canopy where I have a better chance actually of collecting those. And I saw something, if there is anyone eating on the death cap, actually, yes, my favorite species eats on the death cap. That Drosophila flavopenicola is actually capable of eating on deadly poisonous mushrooms. I just saw this on the side here in the chat. Yeah. Let's see.
I do not see any other questions. Anybody else? I think Ty wants to know what he means more is how much does a species range, not necessarily an individual fly.
How do they range? Well, it's very, very difficult. Like every species has different distribution ranges, and they can be spotty and so on. So every species has different ranges, and they overlap also widely. So if I catch something in the northeast, sometimes I find the same species in the northwest as well. Hope that answers the question. And you can also email me. You can share my email address, and I would be happy to give more information about that. Okay.
Yeah, you can put it in the chat when we start the next speaker, if you would. Yes. And then Lissa, I think we have time for one more. Lissa wants to know which particular fruit flies eat the death caps. Is it a specific species or do several species eat it?
It is several species, and they are usually in the Drosophila quinaria group. Half of the quinaria group species, about 10-15 species or so, they eat the death cap. So it's a whole bunch of species, and they are often closely related to each other. Yep.
Great. Thank you, Dr. Werner.